In the previous class, while discussing about the regulation of eukaryotic gene expression, we primarily discussed about once the transcription is over, how does the messenger RNA, what kind of post-transcriptional modifications the messenger RNA undergo. And what I basically told you in the last class is that there is what is called as a both co-transcriptional as well as post-transcriptional modifications that the pre-messenger RNA undergoes during transcription or after transcription. And we discussed two such co-transcriptional processing mechanisms, namely mRNA capping and mRNA splicing. Now, the third most important co or post-transcriptional modification that a pre-messenger RNA undergoes is polyadenylation. So, today we will discuss about the mechanism by which a poly A tail is added to a messenger RNA and how again RNA polymerase II plays a very important role in this event as well. So, today's topic we are going to talk about is co-transcriptional and post-transcriptional modification of pre-messenger RNA part 2. The part 1 we discussed about mRNA capping and mRNA splicing, and in part 2 we are going to discuss about polyadenylation. How does messenger RNA get polyadenylated, what are the enzymes that are involved, and how RNA polymerase II plays a very important role. This is a cartoon I have drawn just to stay in your memory what we are going to talk about. Once the pre-messenger RNA is made, which contains both introns and exons, a cap is added. That is one of the first co or post-transcriptional modifications a pre-messenger RNA undergoes, and we discussed how clearly the RNA polymerase II carboxy-terminal domain plays a very important role in the addition of this cap. And then we also discussed about how the splicing machinery removes the introns and joins the exon together, and even the splicing is highly interconnected with the RNA polymerase II, and again RNA polymerase II plays a very important role, especially the carboxy-terminal domain. It more or less acts like a launching pad for the assembly of both capping factors as well as the splicing factors, and as a result both splicing and capping are carried out simultaneously as the RNA transcription takes place. So, what we will discuss today is about the third important co or post transcriptional event, namely the polyadenylation signal. That is what I depicted here, how a series of A residues are added to the messenger RNA so that you get a poly A tail, and this is what I have shown. So, what we will discuss today is that what is the mechanism by which polyadenylation takes place and a poly A tail is added to the pre-messenger RNA during as well as once the transcription is over. Now, the polyadenylation is a very unique feature of eukaryotic messenger RNAs. It does not take place in prokaryotic species, and not all messenger RNAs are polyadenylated, although majority of them undergo polyadenylation. But there are a small population of RNAs, for example histone messenger RNAs, they do not undergo polyadenylation. And I also discussed in the previous class that this addition of poly A tail has actually given a very important unique research tool to separate out messenger RNA molecules from ribosomal RNA and transfer RNA molecule. So, what you basically do, if you want to isolate messenger RNA from a cell, from a eukaryotic tissue or from eukaryotic cells, you isolate total RNA and then simply take this total RNA and pass it through what is called as an oligo-DT cellulose column, where cellulose is coupled to a series of T residues, and because the messenger RNA alone contains A residues, the A will base pair with T, and only the messenger RNA will be retained on the oligo-DT affinity column, whereas the ribosomal RNA and transfer RNA pass through, and this is how you can specifically enrich or you can specifically isolate polyadenylated messenger RNA from total RNA of eukaryotic cell extracts. So, the presence of poly A tail has actually given a unique tool for purifying polyadenylated messenger RNA from total RNA in eukaryotic cells. Now, let us see how exactly the polyadenylation takes place in the case of eukaryotic cells. The polyadenylation of messenger RNA basically takes place in two steps: recognition of polyadenylation signal and endonucleotic cleavage of messenger RNA precursor. This is the first step, because somewhere there has to be a signal to tell that this is the place where a poly A tail has to be added. So, there has to be what is called as a polyadenylation signal. So, we will now analyze and see what is this polyadenylation signal. Is it conserved among all messenger RNAs or is it different between different messenger RNAs? And once the machinery involved in polyadenylation recognizes that this is the polyadenylation signal, then the RNA is cleaved downstream of this polyadenylation signal, and then another enzyme machinery takes over, which actually adds poly A at the 3' end of the cleaved RNA. So, there are two important events of polyadenylation. One is recognition of the polyadenylation signal and cleavage of the pre-messenger RNA, and once that is over, a series of A residues are added by two enzymes called poly A polymerase and poly A binding protein 2. Now, there are three protein factors which play a very important role in the recognition of the polyadylation signal and endonucleotide cleavage of the pre-messenger RNA, and they are called as CPSF, which is cleavage polyadylation specificity factor, cleavage stimulation factor or CSTF, and there are two cleavage factors called CF1 and CF2. These three are involved in the first step of the polyadylation, and once these three enzymes do their job and cut the pre-messenger RNA, two enzymes called polymerase and polybinding protein actually add a poly-A tail so that you get a polyadenylated messenger RNA species. Let us now take a very brief look at what are these enzymes, what exactly they do. As I mentioned earlier, CPCS is nothing but cleavage and polyadylation specificity factor. It actually is the one which recognizes the poly-A signal, and in majority of the eukaryotic messenger RNAs, the polyadylation signal is highly conserved, and it is a sequence called AAU-AAA. Repeat, AAU-AAA. This is the sequence called polyadylation signal, and this is the signal that is recognized by the CPCF, and it is required for both cleavage as well as polyadylation. So, once the transcription is over, once the RNA polymerase transcribe this poly-A signal, and this poly-A signal present in the RNA is immediately recognized by the CPCSF, and then it recognizes that the transcription has to stop here, and it cleaves the messenger RNA and adds a poly-A tail. And there are two proteins involved in the cleavage of the, once the CPCF recognizes the AAU-AAA sequence, there are two proteins called CF1 and CF2 which are actually involved in the cleavage process. And then is what is called the CSTF, cleavage stimulating factor, which is actually required for cleavage, and it binds to a region called GU/U region, which is immediately downstream of the AAU-AAA region. And then once all these events are over, two enzymes called poly-A polymerase and poly-A binding protein 2 play an important role in addition of the A residues to the end of the RNA. Initially, the poly-A polymerase is involved in the polyadylation process. It adds about 10 to 20 residues of A, and then the poly-A binding protein takes over, and it adds up to 100 residues of A to get a completely polyadenylated messenger RNA. Now let us now look at the sequence which plays a very important role in the polyadenylation, namely the polyadenylation signal. The question we ask is that since there are a number of protein coding genes, there must be a number of messenger RNA synthesized not only in a given cell type but in various tissues and also in different eukaryotic species. The question people ask is that is the machinery that is involved in polyadenylation, does it recognize the same polyadenylation signal? Is it conserved? Just like we have Tata box which is conserved in most of the eukaryotic promoters, we are asked, people start asking the question, are there conserved polyadenylation signal? So when they analyzed a number of eukaryotic messenger RNA sequences, they found out that at the 3' end of the RNA, in majority of the cases, this is the sequence that is highly conserved. You can see 98 genes in 98 out of 100, that is 98% of the genes, the first residue is A. Similarly, 96% of the genes, the first residue is A, the next residue is A, and so on and so forth. So in majority of the genes, this is the signal that that is highly conserved at the 3' end of the messenger RNA. To actually find out whether this sequence really has any role in polyadenylation signal, people did what is called some mutagenesis experiments. So you can see, if you look at the efficiency of polyadenylation of a normal messenger RNA containing AAU-AAA sequence as 100%, you see if you now mutate the second A residues to U, the efficiency dropped down to more than 60%. Similarly, if you now mutate the last A residue to U, there is a dramatic drop in the polyadenylation efficiency. Similarly, mutation of A to G also results in dramatic decrease in the efficiency of polyadenylation, clearly indicating that this sequence AAU-AAA is very essential for efficient polyadenylation of eukaryotic messenger RNA. And as we go along with it, we realize that it is one of the polyadylation proteins binding factors actually recognizes the sequence, and this is what is the first signal for polyadylation of messenger RNA. So remember, a signal called AAU-AAA, we call it the polyadenylation signal, is highly conserved in majority of the eukaryotic messenger RNAs, and this signal is very essential for efficient polyadenylation of eukaryotic messenger RNAs. Now let us briefly discuss about the mechanism by which polyadenylation takes place. Like I told you, the cis-acting elements which are involved in polyadenylation are this signal called AAU-AAA sequence and also a adjacent sequence called GU/U region. And these are the cis-acting sequences which are recognized by specific transacting factors. The first step in this polyadenylation is recruitment of 3 proteins the cleavage and polyadenylation specificity factor CPCSF, the cleavage stimulation factor 1 and CSTF for the the cleavage stimulation factor and the cleavage factor 1. These proteins come and they recognize the AAUA sequence as the GUE sequence, and they bind and they are recruited to the cis acting elements of the messenger RNA, which these elements are present in 3' end. And once these assemble and recognize the poly A sequence, the cleavage immediately about 20 to 30 base pairs downstream of the polyadylation signal, cleavage takes place. And after the cleavage takes place, of these 3 proteins, the CPSF remains bound and a new protein called the poly A polymerase joins, and this poly A polymerase, which I have shown as a red ball here, now adds about 10 residues of A residues. Okay. And another enzyme called the poly A binding protein takes over and now elongates further and adds up to 100 or 200 A residues further. So, this is in a nutshell the mechanism by which polyadenylation of messenger RNAs takes place in the case of eukaryotic cells. Okay. So, you can see here, first is the recognition of the polyadylation signal, which consists of the AAU AAA by 3 polyadylation factors, the CPCSF, CF1 and CSTF, and after this about 20 to 30 base pairs downstream of the signal, the RNA is cleaved, and once the cleavage is over, the poly A polymerase comes and joins the RNA, it adds about 10 A residues, and then the poly A binding protein joins and then expands or extends the A further up to 200 residues. So, this is the mechanism by which polyadenylation takes place. Now, the important point that I would like to discuss, like the way we discussed in the previous lectures, is that RNA polymerase II not only plays an important role in initiation of transcription, but it also plays a very important role in mRNA capping, in splicing, and today we will tell you it also plays a very important role in polyadenylation of messenger RNA. This is the message I want to convey. So, most of the people think the only job of RNA polymerase is to initiate transcription and make RNA, but what is very important, the take home message I want to give at the end of this previous lecture and this lecture is that RNA polymerase II plays a very important role not only in the RNA synthesis, but also in RNA processing, pre-mRNA processing, and especially the carboxyl terminal domain of the largest subunit of RNA polymerase II is very essential for all these 3 post-transcriptional or more appropriately co-transcriptional processes to take place, namely capping, splicing and polyadenylation. And what is the evidence? Why do you think that RNA polymerase II plays a very important role in this? The evidence is that RNAs transcribed by a CTD truncated RNA polymerase II were found to be not efficiently polyadenylated in transiently transfected cell. So, if we have cells which are expressing normal RNA polymerase II with its normal carboxyl terminal domain, and if you have cells in which the RNA polymerase carboxyl terminal domain is truncated, and then if you isolate RNA from these 2 different cell types and look at the efficiency of polyadenylation, in the cells expressing the normal RNA polymerase, the mRNAs are very efficiently polyadenylated, whereas the cells which are expressing a truncated or a deleted form of carboxyl terminal domain, you do not see very efficient polyadenylation, clearly indicating that the carboxyl terminal domain of RNA polymerase is playing a very important role for polyadenylation of messenger RNAs. In fact, it was shown that the 2 factors involved in polyadenylation, which we just discussed in the previous slide, namely the CPSF and CSTF, is actually present in the unfractionated nuclear extracts. They could actually bind to GST-CTD. Now, what is the GST-CTD? Now, if you want to study protein-protein interactions, what you do is, suppose I would like to know, are any of the proteins involved in polyadenylation, are they actually interacting with the RNA polymerase II carboxyl terminal domain? So, what do? I take the carboxyl terminal domain of RNA polymerase II and fuse it to glutathione S transferase. Okay. So, I have a glutathione S transferase C-terminal domain fusion protein, and this can be now made in large amounts in E. coli cells, and since the glutathione S transferase has very high affinity for glutathione, if you now take this whole cell extract containing the glutathione S transferase CTD fusion protein, and you can bind this protein specifically to a glutathione affinity column. Okay. And now you take nuclear extracts which contain all kinds of proteins, and if you pass this nuclear extract to this glutathione affinity column, all those proteins which interact with this carboxyl terminal domain of RNA polymerase II will be retained on the column, and then you can elute these proteins and identify this protein either by N-terminal sequencing of protein or by mass spec and so on and so forth. In fact, this is one of the techniques by which people have actually identified what kind of proteins are actually intact in the RNA polymerase carboxy-terminal domain, and when they actually did these kinds of experiments, they found two of the proteins involved in polyadenylation, namely CPCS and CFTFs, can actually be retained on GST affinity columns, which contains the GST CTD. So, the control is only the GST, whereas the actual protein will be GST CTD, and proteins which are retained only on a GST CTD column but not on a GST column indicates that it is the CTD of RNA polymerase II which is interacting with these proteins. So, when these kind of experiments were done, they actually found both two of the proteins involved in polyadenylation, namely CPCS and CSTS, can be retained on GST CTD columns, indicating that these proteins actually interact with the carboxy-terminal domain of RNA polymerase II. These kinds of biochemical experiments coupled to the in vivo experiments which I told you earlier, that if you truncate CTD and in such cells the efficiency of polyadenylation is very low, these kinds of experiments clearly indicated that the RNA polymerase II C-terminal domain is probably very essential for the polyadenylation to take place. But and see, these kind of experiments were also done in the previous cases, where people have actually shown enzymes involved in mRNA capping, enzymes involved in RNA splicing, they also interact with RNA polymerase II C-terminal domain, and in fact phosphorylated forms of RNA polymerase CTD are more efficient in promoting mRNA capping as well as mRNA splicing. We discussed this aspect in detail in the previous class, but the important distinction between the role of or the interactions of RNA polymerase II CTD between mRNA capping enzymes and splicing enzymes and the interaction of RNA polymerase II CTD with polyadylation machinery is that in the previous two cases, the phosphorylation status of RNA polymerase II played a very important role both in the interactions of the splicing machinery as well as capping machinery with the RNA polymerase II CTD phosphorylation status. But in the case of polyadylation, phosphorylation of CTD does not seem to play a very important role in the interaction with the polyadenylation machinery or proteins involved in polyadenylation. So, unlike capping and splicing, phosphorylation status of CTD was not found to affect interaction of the CPCF and CSTS with the RNA polymerase II CTD. So, all these results clearly indicated that the polymerase II carboxy-terminal domain plays a very important role in polyadenylation. It does help to recruit polyadenylation factors to the site of RNA polymerase II transcription, increasing their local concentration and thereby facilitating efficient processing or efficient polyadenylation. I have given two references where one in Tips and one in Nature. This is the time during the late 90s, late 80s and early 90s, that is the time a lot of effort went on to identify polyadenylation mechanisms and understand the factors involved in this, and people have actually developed in vitro systems. Just like we have a cell-free transcription system, people developed cell-free systems for demonstrating mRNA capping, cell-free systems for demonstrating splicing, and also cell-free systems for studying polyadenylation. So, where you can actually purify factors and put them in a test tube and actually demonstrate polyadenylation can actually takes place. And in fact, using such kind of experiments, people have actually shown if you have extracts in which the RNA polymerase II CTD or RNA polymerase II is depleted, you find the polyadenylation efficiency is very low, and if you now put back RNA polymerase II, you see the polyadenylation now takes place very well. So, both in vivo experiments as well as biochemical experiments involving protein studies as well as cell-free polyadenylation systems demonstrated that the carboxy-terminal domain of RNA polymerase II plays a very important role in the efficient polyadenylation process. Now, how and when do poly A factors associate with the RNA polymerase? Now, people thought, as you can see, polyadenylation is almost the last step in messenger RNA synthesis, because once the RNA is made, then it gets capped, then it gets spliced, and finally the RNA gets cleaved and poly A is added. So, people thought proteins which are involved in polyadenylation probably has nothing to do with RNA polymerase, and even if they interact with RNA polymerase, it must be happening at towards the fag end of the transcription process. But very surprisingly, people found that even at the stage of transcription initiation itself, many of the proteins involved in polyadenylations actually interact with the RNA polymerase II, and they remain bound to the part of the pre-initiation complex. Okay? So, this was the very unexpected answer, where they found that the enzymes involved in polyadenylation actually associated with pre-initiation complex, and they are actually present in the promoter itself. So, although polyadenylation is the last step, almost the last step in the mRNA processing, the enzymes involved in polyadenylation seem to be associated with the promoter itself. And some of the experimental evidences are shown here. For example, while studying the general transcription factor TF2D, it was found that an extensively purified preparation contained a good yield of at least 3 to 4 subunits of CpCF. TF2D, which so far we thought is involved only in transcription initiation, and its job is to assemble the pre-initiation complex and bring the recruit the RNA polymerase to the promoter. The TF2D also seems to be interacting with CpSF, which is a polyadenylation factor. So at least some of the poly factors appear to associate very early during RNA synthesis and during the transcription initiation step itself, and they remain associated with RNA polymerase throughout the process of transcription elongation. Okay. So the crux of what this slide actually tells you is that although polyadenylation is the last step in pre-mRNA processing, the factors involved, some of the factors involved in polyadenylation seem to be associated with the RNA polymerase and the pre-initiation complex at the stage of transcription initiation itself, and right from the beginning they remain associated with the RNA polymerase throughout the transcription process. Now, what is the biochemical evidence demonstrating a more direct role for RNA polymerase II in polyadenylation? This is where I discussed just a little while ago. If you now take the purified RNA polymerase II and RNA polymerase II, both were found to activate the first step in polyadenylation. Remember, the difference between polymerase IIA and O is that one is a non-phosphorylated form and another is a phosphorylated form. As I mentioned previously, for the interaction of the poly factors with RNA polymerase II, phosphorylation does not seem to play an important role. So, whether the CTD is phosphorylated or not, the RNA polymerase II efficiently interacts with poly factors, and therefore both of them are capable of promoting polyadenylation in a reconstituted system containing polyadenylation factors and RNA polymerase II. So, you have a cell-free transcription system which contains all the factors which I just mentioned previously, so that you have a cell-free transcription system where if you now add a pre-messenger RNA, it is capable of getting polyadenylated. Now, in such a system, if you now add an RNA polymerase II with its carboxy-terminal domain, the efficiency of polyadenylation is dramatically enhanced, clearly indicating that the polymerase II, especially the carboxy-terminal domain, plays a very, very important role. And both unphosphorylated and hyperphosphorylated GST-CTD proteins activated the cleavage just as efficiently as the RNA polymerase II, although in this case the hyperphosphorylated CTD was little bit more active than the non-phosphorylated form. Some experiment did suggest that there is some difference in the whether the CTD is phosphorylated or non-phosphorylated. And the 3' cleavage in nucleic acids could be inhibited by immunodepletion of RNA polymerase II and can be rescued back by adding purified enzymes. So, all these studies I am telling you because during the late 80s and early 90s, a lot of effort went into really understand that the RNA polymerase II, in addition to having a role in transcription initiation, also plays a very important role in other subsequent steps of RNA processing, namely capping, splicing and polyadenylation. And since this is against the dogma, nobody thought enzymes which are involved in polyadenylation could be assembled at the time of the pre-initiation complex itself in the promoter, and RNA polymerase to actually be interacting with some of these factors. These were all not unimaginable in those days. So, to convince people that yes, all these things are happening together and the RNA polymerase II CTD playing a very important role, a number of experiments had to be done. For example, in the last step what I said, you actually take an extract, nuclear extract, which is capable of polyadenylation, and in such an extract, if you now immunodeplete, that means you pass this extract through a column which contains antibody against the carboxy-terminal domain of RNA polymerase II, so that the RNA polymerase II along with its CTD can be depleted. And in such extracts they found the polyadenylation does not take place efficiently. To actually show that you have only depleted the RNA polymerase II and its CTD, now if you add back purified RNA polymerase along with CTD, now there is a dramatic increase in the polyadenylation efficiency, clearly indicating that the RNA polymerase II and its CTD is playing a very, very important role in enhancing the efficiency of polyadenylation, and they are very essential for polyadenylation to take place. So, all these kinds of experiments clearly suggested that the carboxy-terminal domain of RNA polymerase II participates directly in the formation of a stable catalytically active processing complex through direct interaction with the polyadenylation factors. A person called James Manley played a very, very important role. He has published a number of papers to study polyadenylation factors, mechanism of polyadenylation, development of cell-free transcription system and so on. I have just shown one of the key papers which was published in late 1990s to demonstrate the link between RNA polymerase II and polyadenylation. I have now shown a cartoon here which actually demonstrates all that we discussed in the last two classes. Basically, this cartoon, which was actually taken from a paper published in BBA in 2002, it's a nice review. I have given the reference also here, and this actually tells you how transcription is coupled to RNA processing. So, if you now look at this, what basically this cartoon tells you is that we now have the pre-initiation complex form. This is the RNA polymerase II, and you have all the general transcription factors like TF2A, B, D, and so on and so forth. But what was surprising is that the purple color, what I have shown here, is the polyadenylation factor. Now, those days, especially in the early 90s, nobody thought an enzyme CPCF which is involved in polyadenylation is actually is part of a pre-initiation complex that at the time of transcription initiation. This was a very surprising finding. So, in addition to the general transcription factors in RNA polymerase II, a factor which is involved in the last step of transcription RNA processing also is associated at the beginning of the transcription itself. So, throughout the transcription, this CPSF remains bound to the RNA polymerase II, and it is only when the RNA polymerase passes through the AAA TAA signal that this poly-A factors now jump into the RNA and then cleave the RNA. Till then, they remain associated with the RNA polymerase II CTD. Okay, now let us see what happens. Once the pre-initiation complex is formed, the TF2H, the transcriptionation factor, the general transcription factor TF2H now phosphorylates the RNA polymerase II. That is shown in the red asterisk here, and once the RNA polymerase II is phosphorylated, that is the signal for elongation to start. So, the PIC leaves the promoter and starts transcribing, and when the RNA is about 20 nucleotides old, the capping enzyme, all the enzymes for mRNA capping now hop on to the phosphorylated RNA polymerase CTD and start doing the mRNA capping. They add the 5-methyl cytosine cap to the 5' end of the messenger RNA. And once the capping is over, then enzymes involved in splicing now hop on to the RNA polymerase II CTD, and they carry out this splicing process, and these 3 steps actually show various splicing taking place and the introns getting spliced out. Basically, what I am telling you is that both the capping machinery as well as splicing machinery are happening as the transcription is proceeding. All this RNA processing does not. So, the earlier concept that mRNA capping and splicing as post-transcriptional has now paved way to being defined as co-transcriptional. This is the message I want to convey. mRNA capping and at least some of the processing, some of the splicing reactions are co-transcriptional rather than post-transcriptional processes. So, and once the splicing, once the capping is over and once the splicing is taking place, and once the RNA polymerase traverses the poly-A signal in the gene and the polyA RNA is synthesized, the capping enzyme which was actually interacting with the RNA polymerase CTD now hops on to the AAUA sequence of the RNA and then immediately cleaves, and then the poly-A binding protein now adds the poly residues, and you have now got the poly-A polyadenylated messenger RNA coming out into the nucleus. Now, there are also 2 important steps which I have not discussed during the RNA processing steps because it is a little bit advanced and I do not want to confuse the readers. And there are also regulations taking place at the level of transcription elongation. So far, we have discussed regulation at the level of transcription initiation, and also during RNA processing, how alternate splicing can lead to more than one messenger RNA and so on and so forth. But transcription regulation can also take place at the level of transcription elongation. Okay, but maybe I will just show 1 or 2 slides to actually show the role of elongation factors in regulating transcription. And there are also regulation taking place at the level of transcription termination. For example, a protein called FCP1 plays a very important role, and this is the one that actually now dephosphorylates the carbox terminal domain, and therefore the RNA polymerase can be now recycled back and can again go back to the promoter, again interact with general transcription factor and start one more round of transcription. So, this is what is called the transcription cycle. You have initiation, elongation and termination, and the message I wanted to give you is that the pre-mRNA processing, which involves capping, splicing and polyadenylation, all these 3 are coupled to transcription and they are all highly integrated, and all the 3 take place in a very harmonious manner, and RNA polymerase II, especially the C-terminal domain, plays a very important role not only in transcription initiation but also in elongation, capping, splicing and polyadenylation. So, this is just the legend for the previous figure. You can just go through this. This is just the crux of what I told you that the 2 of the enzymes, proteins involved in polyadenylation, namely the CPCF and CSTS, they hop on to the carbox terminal domain of the RNA polymerase II and remain associated with the carbox terminal domain throughout the transcription process. but once the RNA polymerase II holoenzyme crosses the polyadylation sequence, and once the RNA gets transcribed, and once you have the polyadenylated messenger RNA coming out of the RNA polymerase, immediately these enzymes leave the RNA polymerase and hop onto the RNA, and immediately they cleave the messenger RNA, and then the poly A tail is added and polyadenylated messenger RNA is released. So this is the mechanism by which polyadenylation takes place, and the interactions between machinery of polyadenylation and RNA polymerase to C-terminal domain. I just given a number of papers which describe the major advances that took place in the discovery of or deciphering the mechanism of polyadenylation, especially the coupling of or the role of RNA polymerase to C-terminal domain in polyadenylation. So you can just go through some of these papers and then read a little bit more about and as I told you, James Manley has published a number of papers during the late 90s and early 2000 to decipher or understand the mechanism of polyadenylation. So what was very surprising is that the RNA polymerase II, especially the CTD, the C-terminal domain, participates in mRNA processing was a kind of unexpected, and it was highly controversial. In the initial stages, nobody believed that the RNA polymerase II, which is actually involved in transcription initiation, may be having a role in the subsequent steps of RNA processing. So it took a lot of time and lot of experimental evidence to convince, biochemical experiments, in vivo experiments, cell-free studies, to actually convince that all these processes are integrated. So if you do not have RNA polymerase CTD, not only transcription initiation affected, capping is affected, splicing is affected, polyadenylation is affected, and to convince people that is real, a lot of experiment needs to be done during the late 90s and early 2000. So the bottom line, what came out on this lecture and the previous lecture is that a mature eukaryotic messenger RNA contains a 5' cap, a 5' methyl cytosine at the 5' end. So it is capped, and then all the introns are removed, and then it contains the poly A tail at the 3' end. And there are three major RNA processing mechanisms that we have discussed so far. A cap is added to the 5' end of the nascent transcript when the mRNA is about 20 nucleotide long. So capping does not wait till the entire transcription is completed. Even as the RNA is coming out of the RNA polymerase elongation complex, even as long as even the RNA is just 20 nucleotide old, the capping takes place. And as the RNA synthesis continues, the spliceosomes are assembled onto the introns as transcription continues. Splicing is usually it is not completed until the nascent transcript is relayed. So unlike capping, so I can actually call capping as a co-transcriptional process, but splicing is probably both co-transcription as well as post-transcriptional process. So while capping is completed even before the RNA synthesis is completed, splicing is initiated co-transcriptionally and completed post-transcriptionally. This is the message I want to give. So the two distinctions between capping and splicing is, capping is done in a co-transcriptional manner, is co-transcriptional, whereas splicing is initiated co-transcriptionally and completed post-transcriptionally. Now, shortly after the RNA polymerase II passes the polyadylation signal, the 3' end of the transcript is generated by cleavage, and then polyadylation takes place. So three RNA processing mechanisms, capping, splicing and polyadylation, are either co-transcriptional or co-transcriptional as well as post-transcriptional, and RNA polymerase II C-terminal domain plays a very important role in all the three processes. This is a cartoon for you to easily remember. You have RNA polymerase and you have the RNA polymerase CTD, and the RNA polymerase II CTD is involved not only in initiation of transcription, elongation, capping, polyadylation, splicing. So this is the guy who is conducting RNA polymerase II CTD is the one who is conducting an orchestra, and who are the players? These are all the people who are playing various instruments, and all these instruments are being controlled by one person who conducts the orchestra. And in the same way, RNA polymerase II CTD not only controls elongation initiation, it also controls elongation, capping, polyadylation as well as splicing. And all these factors which are involved, many of these factors which are in all these processes, they interact with either unphosphorylated or phosphorylated form of C-terminal domain of RNA polymerase II. So this is the take-home message of this lecture and the previous lecture, that mRNA processing is a co-transcriptional and post-transcriptional mechanism, and it happens as the transcription proceeds, and RNA polymerase II plays a very important role in not only initiation but also in the subsequent RNA processing events. So, throughout the transcription cycle, the RNA polymerase II carbox terminal domain undergoes a variety of covalent and structural modifications, which can in turn modulate the interactions and functions of other factors involved not only in transcription initiation, but also elongation, termination, capping and splicing. So, this whole thing is a very dynamic process. So, many residues of CTD are getting phosphorylated, dephosphorylated, depending upon which residue is getting phosphorylated, which residue is getting dephosphorylated. It interacts either with capping enzymes or splicing enzymes or with polyadenylation enzymes, and the whole thing is a very dynamic process. So, throughout the process of transcription elongation and termination, the CTD is undergoing dynamic changes of phosphorylation, dephosphorylation and differential protein-protein interactions. So, inside the nucleus there is a harmonic integration of transcriptional and post-transcriptional activities, many of which were once considered to be functionally isolated in the cell. So, the take home message I want to give from this lecture is that in the early 80s or late 80s, people thought initiation is a different event, and once the transcription is over, then the mRNA processing takes place. Capping is added separately, splicing is added separately and polyadenylation acts separately, and this all happens in different places in the nucleus. But what we now know today is that these are all highly integrated. They all happen in both in space as well as time, they happen together. Okay, they are all highly linked, and all the proteins or enzymes involved, all these processes, they interact with each other and they are all highly linked. This is one of the major findings of research coming out of studying transcription initiation as well as post-transcriptional processing of messenger RNA. So, a number of experiments, as I said, it took a lot of time or a lot of effort to actually prove this point that the RNA polymerase II, especially the C-terminal domain, plays a very important role in the co-transcriptional processes, and I am just listing one or two experiments so that it registers in your mind. For example, here is a cartoon. How do we demonstrate that a RNA polymerase II, especially the C-terminal domain, really has a role in mRNA capping? If somebody asks a question, what do? You now take cell types. There are two cell types, one expressing normal RNA polymerase with C-terminal domain, another expressing a truncated RNA polymerase II without C-terminal domain. And if you now look at the messenger RNAs which are being synthesized, and you find that only in the cells which contains the normal RNA polymerase with its complete C-terminal domain, messenger RNAs are capped, whereas cells expressing RNA polymerase II in which the CTD is not there, RNA is made, but the RNA is not efficiently capped. And what people later found with these kinds of experiments, not only capping is getting affected in these kinds of cells where the CTD is either truncated or mutated, other events like splicing, polyadenylation is also getting affected. So, using these kinds of in vivo experiments, people have clearly demonstrated that the C-terminal domain of RNA polymerase II plays a very important role in pre-mRNA processing. See, for example, the phosphorylation of serine 5 of C-terminal domain is essential for the recruitment of capping enzyme to polymerase II complex, as well as for the allosteric modulation of the guanylyl transferation of the capping enzyme. Now, the important point I want to mention is that not only RNA polymerase CTD acts like a launching pad for proteins to come, do their job and go back. Okay, like for example, the capping enzymes come, sit on the RNA polymerase II, cap the messenger RNA and go back. The splicing machinery again come back, sit on the RNA polymerase II. It's more like acts like a launching pad for all these enzymes, but it's not just that. It also has an active catalytic role. For example, in the case of capping, the C-terminal domain not only is just serving as a platform for the assembly of these factors, but it actively enhances the rate of mRNA capping. So, it actually modulates the activity of enzymes which are involved in mRNA capping. So, it has a very active role in all these post-transcriptional processes. And mRNA capping is co-transcriptional. And mRNA splicing and polyadenylation are also co-transcriptional. Again, very elegant experiments were done. For example, here is a paper in 1993 where a person called Greenleaf actually suggested that the negatively charged hyperphosphorylated CTD could act as a ionic scaffold for the recruitment of arginine-rich proteins called as SR proteins, which are actually involved in splicing and phosphorylation states of CTD, and the serine residues of the RS domains of SR proteins might regulate these interactions. So, this paper in 1993, a review in Tips, actually talks about how RNA polymerase II can play a very important role in splicing, and depending on the phosphorylation status of the RNA polymerase CTD as well as the phosphorylation status of the serine rich proteins or the SR proteins which are involved in splicing, the splicing may be actually regulated by RNA polymerase II. Again, the experimental evidence for physical linkage between transcription and splicing was actually demonstrated first by Chabot et al. In 1995, who actually showed that RNA polymerase II, spliceosomal snRNPs and splicing intermediates could be co-immunoprecipitated using a monoclonal antibody direct against the phosphorylated form of CTD. So, if you now take cell extracts which contain RNA polymerase II with its CTD and other factors, and if you now have an antibody which is specifically raised against the C-terminal domain of RNA polymerase II, and if you do immunoprecipitation, not only the RNA polymerase II CTD comes, along with that you can also pull down proteins involved in splicing, indicating that many of the proteins involved in splicing actually interact with the phosphorylated form of RNA polymerase II. So, these kinds of protein-protein interaction studies clearly demonstrated there is linking between splicing and RNA polymerase II. So, as I said, the take-home message is that splicing polyadenylations begin co-transcriptionally and are completed post-transcriptionally, whereas mRNA capping is primarily co-transcriptional. Also, other papers which actually demonstrate, these are the two papers which actually demonstrate for the first time that the carboxy-terminal domain of RNA polymerase II subunit is actually required for efficient RNA processing. One published in Nature 1997, the C-terminal domain of RNA polymerase II couples mRNA processing to transcription, a very important paper where they have actually shown splicing, processing of 3' and termination of transcription downstream of the polypeptides all are inhibited when you have cells expressing truncated form of C-terminal domain. So, if you read this paper, it becomes very clear what kind of experiments they have actually done to demonstrate that if you truncate the C-terminal domain of CTD of RNA polymerase II, splicing as well as 3' end formation as well as polyadenylation all are affected, indicating that the CTD plays a very important role. And they have also actually done experiments to actually demonstrate that cleavage polyadenylation factors like the CPCF and CSTF actually binds to CTD using affinity chromatography studies, and they actually co-purify with RNA polymerase II as a high protein complex, high molecular weight protein complex. So, there is an association. So, these authors based on their experiments actually demonstrated, proposed in 1997, that there is an association with the carboxy-terminal domain of RNA polymerase II and 3' processing factors, suggesting that inside the nucleus an mRNA factory exists. And this mRNA factory carries out not only transcription but also splicing, cleavage, polyadenylation all together. So, these are all co-transcriptional and they don't happen in an isolated manner inside nucleus, but they are all highly integrated. So, these are all very important factors that actually are responsible for some of the major advances that took place in the area of RNA processing. I have just shown one or two slides to actually demonstrate that regulation of transcription also takes place at the level of elongation, but I will not elaborate too much on that. Just like we have factors which regulate transcription initiation, splicing, capping and polyadenylation and so on, there are also factors which enhance or inhibit the rate of transcription elongation. All the genes are not elongated at the same time. The RNA polymerase does not make all the RNAs at the same rate, and there are factors in the presence of which RNA polymerase II either the process to RNA polymerase is either high or slow. For example, there are proteins which actually suppress transient pausing and stimulate the rate of transcription elongation of RNA polymerase. These proteins are called TF2F, ALL, elongin and so on and so forth. Similarly, there are factors that reactivate an RNA polymerase molecule which was arrested during transcription, and these are called the TF2S proteins. And there are actually factors that stimulate elongation on chromatin templates called HMG14, HMG17 and FACT and so on and so forth, so that on chromatin templates the rate of transcription elongation can be regulated. And there are a number of other factors. I will not go into the details. Those who are keen actually can study a nice review which appeared in BBA sometime in the year 2002. Now, I am going to spend maybe another 10 minutes to just explain another very important RNA processing mechanism. So far we discussed mRNA capping, splicing and polyadenylation, and we made it very clear these are either co-transcriptional or post-transcriptional modifications. But are there any other RNA processing mechanisms? Are there any other co or post-translational modifications that a pre-messenger RNA undergoes? There are many others, but one important that I would like to mention in the next maybe 5-10 minutes is a mechanism called RNA editing. Okay. Now, RNA, so we discussed mRNA splicing, mRNA capping, mRNA polyadenylation. There is also a mechanism called RNA editing. Now, what is RNA editing? RNA editing is again a co or post-transcriptional modifications of RNA primary sequence from that encoded in the genome through nucleotide deletion, insertion or base modification mechanisms. That means once the pre-messenger RNA is synthesized, either certain bases can be added or certain bases can be deleted or certain bases can be modified. That means the pre-messenger RNA sequence need not be exactly same as the mature messenger RNA because of these modifications. We already know one of the major differences between the pre-messenger RNA sequence and the mature RNA is the mature RNA does not contain introns. In addition to that, there are also certain nucleotides which may be different between the two because of RNA editing. So let us now see how this takes place. Now this process of RNA editing was first discovered in the kinetoplastids, which are single cell protozoa, namely the Trypanosoma brucei, Trypanosoma cruzi, Leishmania, and etc. So this mechanism, this process of RNA editing was first discovered in some of the protozoan parasites. Now what is this RNA editing? There are basically two types of RNA editing that takes place in eukaryotic cells. One is called as the base modification or substitution editing. This is primarily carried out by enzymes called adenosine deaminases. There are two types of base modification which can happen. The adenosine residue in the primary messenger RNA or the messenger RNA can be converted to inosine by deamination reaction. This is primarily seen in viruses, certain human genes, and another important RNA editing that takes place is conversion of cytosine to uracil or uracil to cytosine. This is seen in chloroplast, plant mitochondria, and again certain human genes. So this kind of modifications is called where existing base is either converted into another base, either A is converted to inosine or cytosine is converted to uracil or uracil converted to cytosine. This is called as the base modification mechanism, and these kinds of changes are primarily carried out by a family of enzyme called as adenosine deaminases. The other important method of RNA editing involves a insertion or deletion. Here you are just modifying a base, but here you are actually either inserting or deleting a base. The most common RNA editing that happens is the insertion of U residues or deletion of U residues, primarily seen in the again kinetoplastid protozoans, and in certain other cases or organisms it can also results in the insertion of dinucleotides, or they can actually result in the replacement of one nucleotide by another. So all this involves RNA editing. Base modification, insertion or deletion of nucleotides, and I have just shown one example here. For example, if this is the pre-messenger RNA sequence here, and if it undergoes an insertion editing, what happens? If U residues are inserted, for example, you can see here after the AAAG, two U's are inserted here, and similarly after UG again two U residues are inserted here, and as a result the open reading frame of pre-messenger RNA will be completely different from the open reading frame of the edited messenger RNA. So if you look at the genomic sequence, you will see this, but if you actually sequence the mature RNA and or if you deduce the amino acid nucleotide sequence based on the protein, you will find the deduced nucleotide sequence based on the protein sequence does not match with the genomic DNA sequence because of this RNA editing phenomena. So you conclude that, how do you conclude that the gene sequence is different from the mature RNA sequence? When you deduce the amino acid sequence from the gene, and if it does not match with that of the actual messenger RNA, if you sequence a cDNA and the cDNA sequence does not match with the actual genomic sequence, you conclude that some kind of a co or post-transcriptional modification has taken place. I repeat again, how do you demonstrate that in messenger RNA some kind of a post-translation or co-transcriptional modifications have taken place for a particular gene? If you now sequence the cDNA, because cDNA is primarily made from messenger RNA, and if you sequence the cDNA and if you also sequence the gene from the genomic DNA, and the genomic DNA sequence and the cDNA sequence do not exactly match, one conclusion is that naturally because introns may be spliced, but another could be some kind of an RNA editing might have taken place, and as a result where, for example, if a base modification has taken place, there may be inosine in the cDNA, whereas it may be adenosine in the genomic DNA. Similarly, if you find that certain extra U's are there when you sequence cDNA, where that kind of U's or T's are not there in the genomic DNA, it again means that a post-transcriptional modification has taken place, and an RNA editing is probably the cause why the cDNA sequence is different from that of the genomic DNA sequence. Substitution editing, example as I told here, the most common is what is called as adenosine to inosine modification. RNA editing by members of ADAR, which means adenosine deaminases acting on RNA. ADAR, adenosine deaminases acting on RNA. This family of enzymes convert adenosine into inosine in some of the precursor messenger RNAs. So these are actually known as inosine containing messenger RNAs, and what is the significance of this? There is a very nice review in Nature Biotechnology, and it turns out especially in the as late as 2005 and the early 2000s, people have now realized that many of the transcripts, see, usually after transcription and after RNA processing, the messenger RNA comes out of the nucleus, goes into cytoplasm, gets translated, and a protein is made. This is the common mechanism. But people have found out there are many messenger RNAs which are actually transcribed by RNA polymerase II, but they don't actually leave the nucleus. They stay inside the nucleus. They don't come out. And when they actually found out what they are, they come out only after certain kinds of modifications. So when they've actually found out, analyzed what are these messenger RNAs which are transcribed by RNA polymerase II, but they stay inside the nucleus, they found that many of these RNAs actually contain inosines. So the adenosine to inosine modification is one of the mechanisms by which transcripts are retained in the nucleus. So the A to I RNA editing is actually involved in the retention of messenger RNA inside the nucleus. Such RNAs containing inosine residues are usually not seen in the cytoplasm, they are primarily present in the nucleus. So this is one mechanism by which certain RNA molecules are selectively retained within the nucleus and they don't come into the cytoplasm at all. So this is one of the major significance of this major RNA editing mechanism. I will give you one other very important and very classical example of a substitution editing. There in the previous slide I showed you adenosine to inosine conversion, and that kind of an RNA editing seems to play a very important role in nuclear retention of messenger RNAs. Now here is an example where a cytone is getting converted to U, and as a result it results in the formation of two different proteins. For example, there is a gene that codes for a protein called apolipoprotein B, known as abbreviated as ApoB. Now, if you look at the gene of this apolipoprotein B gene, there are two sequences which are highlighted here. One is CAA, another is TAA. Now, when there is no RNA editing that is taking place, especially this happens in liver, the RNA is transcribed and the RNA is translated, and since TAA is a stop codon, it stops here, and as a result you generate an apolipoprotein B protein which is about 100 amino acids long. So if you look at if you purify apolipoprotein B from liver cells, and you find out that this protein contains 100 amino acids, and it starts from ATG and ends at the stop codon here. Interestingly, in intestine, the same gene, the RNA that is transcribed from the same gene in liver undergoes a C to U editing, the substitution editing, and as a result this particular C is converted into U. So CAA now has become UAA, and you know that UAA is a stop codon. So as a result, when this RNA comes from the cytoplasm, translation does not proceed beyond this point, and therefore you get a truncated protein which is only 48 amino acids. So you can see from the same gene, the gene is the same whether it is a liver or intestine, but the messenger RNA, if this does not undergo RNA editing, it generates an apoprotein protein of 100 amino acids, whereas in another tissue, because the RNA undergoes editing, it generates a protein of just half the size. So you can see how from a single gene you can generate two different proteins by RNA editing mechanism. And I just mentioned here why RNA editing takes place. Only this particular CAA is recognized. There is something called as a Neville nucleotide sequence, which is known as the mooring sequence, which is in and around the CAA, and these flanking nucleotides in and around the CAA is within the apolipoprotein protein B are actually recognized by these proteins involved in RNA editing. In the case of apolipoprotein B, these proteins are called ApoBec1 and ACF1, that is ApoBec1 complementing factor. So these protein factors recognize the 11 base pair sequence in and around CAA, and as a result they bring about RNA editing, especially in this particular region, and therefore a CAA which codes for an amino acid is converted into a stop codon. As a result, translation stops here and you get a truncated protein. From a single gene, by RNA editing you generate two different proteins in two different tissues. So now with genome sequence being out, we now know how many protein coding genes are present and so on and so forth. What now people are now examining is that how many genes actually undergo RNA editing, how many genes undergo splicing, because by these kinds of post-transcriptional mechanisms, although the number of genes may be only 30 or 40,000, you can now realize because of alternate splicing, differential splicing, different RNA editing, the number of transcripts and number of proteins you can generate are many times more than the actual number of genes, because you can generate more than one messenger RNA, you can generate more than one protein by many of these post-transcriptional mechanisms. And for example, systematic identification of abundant adenosine to inosine editing sites in human transcriptome, this was published in 2004, and these people predict that there are something like 12,723 editing sites in 1,637 genes, and they actually experimentally validate 26 such genes, indicating that there are this many genes that undergo RNA editing, and there are this many sites which are potentially can undergo RNA editing. And they offer many new editing sites, and actually experimental evidence as how RNA editing may be a much more common mechanism than people previously thought. In fact, based on the EST and other expression data, it is now predicted that up to 80% of the human genes are believed to undergo produce alternative based messenger RNA. See, when human genome sequence or mouse genome sequence was published, and when people realized that it contains only 30,000 or 50,000 genes, people are surprised. But you can see by these kinds of mechanisms, you can generate lot more proteins and lot more messenger RNAs by all these post-transcription mechanisms, and therefore from one gene you can always generate more than multiple messenger RNAs and multiple proteins. I'll just take 1 minute to explain that. So far, if you have been studying in textbooks, you will realize that a nucleus contains only what is called as a nucleolus. But what studies have now shown is that once people realized splicing, polyadenylation, all these things are coupled to transcription, people have actually identified a number of subnuclear structures. I will not go into the details of this because we will discuss some of this at a later time point in the course. I am just mentioning some of these things here. A number of subnuclear structures have been recognized in the recent studies, and we will discuss many of these things later during the course. There are something called as nuclear speckles, for example, which play a very important role in storage of certain RNA molecules. We will discuss this again later during the course. So, we now come to the conclusion of this particular lecture. Now, this is the 12th in this particular course. So, this lectures from lecture 1 to lecture 12. Primarily, we have focused our attention on the synthesis of messenger RNA. How messenger RNA synthesis is regulated inside the cells. We started from basal transcription factors, promoter elements, we discussed transcription factors, then we discussed the role of chromatin, then we studied epigenetic regulation, then we discussed very intensely the role of chromatin, and then we discussed in the last two classes what kind of processings that mRNA undergoes, and with this one aspect, one module of this course has been completed, and these 12 lectures, if you study, you will get a very good idea about how regulation of RNA polymerase II transcription takes place inside the mammalian cells. So, based on all these 2 lectures, I have listed a number of exercises here. So, if you have really understood all these 12 lectures and have really got a good knowledge about messenger RNA synthesis and regulation, you should be able to answer many of these exercises. I have listed a number of questions here. I suggest you go through many of these questions and then see whether you can answer many of these questions. If you cannot answer, go back to those lectures and read up some of these things and see whether you can answer some of these questions, and this is the way by which you can get a very comprehensive knowledge of how messenger RNA synthesis is regulated inside the cells. I will not go through many of these questions, but you can read up and see whether you are able to answer some of these questions. I think I will stop here.